Today we are going to read about an important enzyme that links the glycolysis and TCA cycles. So that enzyme is pyruvate dehydrogenase complex. As I have already told you, this pyruvate dehydrogenase complex links glycolysis and TCA cycle, and this pyruvate dehydrogenase complex is a mitochondrial enzyme. This mitochondrial enzyme links the glycolysis and TCA cycle. So here what happens, from the glucose after the glycolysis, pyruvate is obtained, and from the pyruvate, if there is anaerobic metabolism, lactate is produced. So pyruvate is converted to acetyl coenzyme by the oxidative decarboxylation, and this is an irreversible enzyme. So this is catalyzed by the multi-enzyme complex that is known as the pyruvate dehydrogenase complex, which is only found in the mitochondria. And the high activities of pyruvate dehydrogenase complex is found in the cardiac muscles and kidney. So again I am going to tell you, from the glucose after glycolysis, pyruvate is obtained, and pyruvate upon action by the pyruvate dehydrogenase complex, acetyl coenzyme A is produced, and from the acetyl coenzyme A, the citric acid cycle is commenced. So this is the one of the main thing you have to know in the pyruvate dehydrogenase complex enzyme. So the sequence of reaction brought about by the different enzyme of pyruvate dehydrogenase complex in association with the coenzyme is shown in this figure. So this is the main thing you have to know, and the net reaction in the pyruvate dehydrogenase complex that is involved is pyruvate plus NAD plus coenzyme A upon the action of the pyruvate dehydrogenase gives acetyl coenzyme A, carbon dioxide, and NADH. So the complex contains 3 enzymes, and it requires 5 cofactors. The 3 enzymes are pyruvate dehydrogenase, dihydrolipoyl transacetylase, and dihydrolipoyl dehydrogenase. These are the 3 enzymes in the pyruvate dehydrogenase complex, and the 5 cofactors that catalyzed these mitochondrial enzymes are thyramine pyrophosphate, which is B1, lipoic acid, coenzyme A, which is B5 or pantothenic acid, FAD, which is B2 riboflavin, and NAD+, which is B3 niacin. So these are the 5 cofactors. You have to know these 5 cofactors because this is the very important cofactors that helps to do the reaction of the pyruvate dehydrogenase complex. So these are the 5 cofactors. I'm again going to repeat. These are the thiamine pyrophosphate, lipoic acid, CoA, FAD, and NAD+. These are the 5 cofactors. Another a comparable enzyme with the pyruvate dehydrogenase complex is alpha ketoglutarate dehydrogenase complex of the citric acid. So alpha ketoglutarate dehydrogenase complex is one of the comparable enzymes of the pyruvate dehydrogenase complex because it uses the same cofactors. It uses same 5 cofactors. The detailed explanation of alpha ketoglutarate dehydrogenase complex, I will discuss that in citric acid cycle. Now let us discuss about the regulation of the pyruvate dehydrogenase complex. In the regulation of the pyruvate dehydrogenase complex, we have to know that the pyruvate dehydrogenase is a good example of the in-product inhibition. Beside this, the pyruvate dehydrogenase is also regulated by the phosphorylation and dephosphorylation. The pyruvate dehydrogenase is active at dephosphorylation stage, while it is inactive in the phosphorylation stage. Pyruvate dehydrogenase phosphate activity is promoted by the calcium and magnesium and insulin. Whereas the pyruvate dehydrogenase is inhibited by the enzyme like NAD+, coenzyme A, and pyruvate. So this is the I said the just jumble words. Forget about that what I have told you, and I will again explain you in simpler form. So pyruvate dehydrogenase complex has 2 forms, phosphorylated form and dephosphorylated form. The pyruvate dehydrogenase complex is inactive in the phosphorylated form, but pyruvate dehydrogenase complex is active in dephosphorylated form. Again, I am going to tell you, pyruvate dehydrogenase complex is inactive in phosphorylated form, whereas the pyruvate dehydrogenase complex is active in dephosphorylated form. That means if the pyruvate dehydrogenase has a phosphate group, then it is inactive. Whereas if the pyruvate dehydrogenase complex, if phosphate group is removed, then the pyruvate dehydrogenase complex become active. So the removal of the phosphate group in the pyruvate dehydrogenase complex is brought about by the pyruvate dehydrogenase phosphatase enzyme. But the addition of the phosphate group in the pyruvate dehydrogenase complex is brought about by the pyruvate dehydrogenase kinase. So upon action of pyruvate dehydrogenase phosphatase on the pyruvate dehydrogenase complex in the phosphorylated form, it becomes dephosphorylated enzyme and the pyruvate dehydrogenase becomes active. So the various substrates that act in the pyruvate dehydrogenase phosphate, which increase the activity of pyruvate dehydrogenase phosphatase, also increase the activity of pyruvate dehydrogenase complex. That means the pyruvate dehydrogenase phosphatase activity is enhanced by the calcium, magnesium and insulin. That means calcium, magnesium, insulin acts on the pyruvate dehydrogenase phosphate. It increases the activity of pyruvate dehydrogenase phosphatase, and pyruvate dehydrogenase phosphatase makes the inactive pyruvate dehydrogenase complex to active pyruvate dehydrogenase complex. So you have to know that. But the another enzyme is that pyruvate dehydrogenase kinase. The pyruvate dehydrogenase kinase helps pyruvate dehydrogenase complex to gain one more phosphate group. It makes active dephosphoenzyme to inactive phosphoenzyme. So the NAD plus, coenzyme A and pyruvate inhibit the pyruvate dehydrogenase kinase. That means there is less conversion of the active dephosphoenzyme to inactive phosphoenzyme. That means NAD plus, coenzyme A and pyruvate enhances the action of pyruvate dehydrogenase complex, whereas ATP, NADH plus, acetyl coenzyme A enhances the activity of the pyruvate dehydrogenase kinase, and it helps to make the active pyruvate dehydrogenase complex enzyme to inactive pyruvate dehydrogenase complex. So overall, I am going to tell you is that calcium, magnesium, insulin, NAD plus, coenzyme A and pyruvate enhances the activity of pyruvate dehydrogenase complex, whereas ATP, NADH and acetyl coenzyme A decreases the action of the pyruvate dehydrogenase complex. So this is the main thing you have to know. So you have also learned about the regulation of the pyruvate dehydrogenase complex. Now let us talk about the arsenic poisoning. The arsenic poisoning inhibits the enzyme pyruvate dehydrogenase by inhibiting the lipoic acid. So after inhibiting the lipoic acid, I told you that for the proper functioning of the pyruvate dehydrogenase complex, one coenzyme is required, that is lipoic acid. If after the arsenic poisoning, what happens? The lipoic acid is inhibited. So the pyruvate dehydrogenase complex cannot work properly, and there is more accumulation of the pyruvate in the tissues. So what are the various clinical features in the arsenic poisoning? There is skin changes, there could be skin cancer, vomiting, diarrhea. In the ECG you see the QT prolongation, and there is garlic breath. So these are the main clinical features you see in arsenic poisoning. So these are the main important things you have to know in the pyruvate dehydrogenase complex. You learn about the significance of the pyruvate dehydrogenase, and knew that the pyruvate dehydrogenase is an important mitochondrial enzyme that links the glycolysis and TCA cycle. You also learn about the reaction of the pyruvate dehydrogenase and what are the various enzyme and cofactors that are needed for the proper functioning of the pyruvate dehydrogenase complex. And you also learn about the regulation of the pyruvate dehydrogenase complex, and also you learn about the arsenic poisoning. So thank you for today. I hope you understood this explanation of the pyruvate dehydrogenase complex. Thank you.